Okay, the next presentation is Martin Harvey. Martin Harvey is an entomologist and biological recorder based at CEH and Biological Record Centre. His main areas of work include the iRecord online recording system, liaison with national recording schemes, and the UK Pollinator Monitoring Scheme, which he's going to be talking about today. As a volunteer, Martin runs the National Soldier Fly and Allies Recording Scheme and is a county moth recorder for Berkshire Vice County. And I can't tell you the number of times that Martin Harvey has bailed me out with iRecord queries at the last minute, minutes before a BioBlitz or something similar. So he's someone who I'm very grateful for his work. So I'd like to pass over to Martin Harvey to talk about the UK Pollinator Monitoring Scheme. Are you there, Martin?
I am indeed. Thanks very much, Simon. Hopefully you can hear me and hopefully you can see the screen now. Okay, let me just arrange the screen so that I can see it. Good. Okay. So yes, I'm going to talk about the UK Pollinator Monitoring Scheme, which we tend to refer to as Poms. And there isn't a specific urban connection here, so I'm not quite following the theme of the morning as closely as some of the other talks this morning. But pollinator monitoring can take place in pretty much anywhere where you might find pollinators, and we certainly do have counts coming in from urban areas, so we'll touch on that a little bit later. So what is the UK Pollinator Monitoring Scheme? The background to the Pollinator Monitoring Scheme really dates back to about 2014 when DEFRA published the government national pollinator strategy, and there are similar strategies in existence for all the UK countries now. And those strategies picked up on a whole range of issues to do with pollinators. As I'm sure everybody is aware, there's been an awful lot of concern about pollinator potential declines and what we can do to conserve them and how we can maintain those really important ecosystem services that pollinators provide for us. And the strategy includes lots of actions towards those ends, but also recognised that it was really important to have some evidence alongside the other work that was going on to track what is actually happening to pollinators and to try and get a better idea of what changes are occurring. And of course, we do have an awful lot of recording data in the UK on groups such as bees and hoverflies and many of the other pollinating insects. But the recording scheme data is fantastic for tracking changes in distribution and looking at which species are increasing and declining in that sense. But it's been really hard to get systematic information that brings in the abundance of those pollinators and whether the actual number of individual insects seems to be changing and how that relates to the changes in species distribution. So Poms has set up 2 new surveys, but we are very much working with the existing recording schemes and trying to bring together the strengths of the data that's collected via those recording schemes with these new surveys that try to focus a little bit more on the abundance of these insects. And I think one of the other things that Poms is trying to do is to focus not just on the well-known pollinators. People tend to immediately think of bees when you talk about pollinators, and perhaps hoverflies as well. But there are an enormous range of different insects and different insect groups that are involved in pollination. I think Stephen Falk has estimated that it could be as many as 6,000 different species across all the different groups. And that's part of what we are trying to focus across a wide range of groups with these surveys. So the 2 surveys that Poms runs, the first one is known as the FIT count, the flower insect timed count. And this is one that anybody can do pretty much anywhere as long as you can find some flowers growing. A lot of them do get done within gardens or parks, but potentially even a flower pot on a windowsill outside, of course, would enable you to carry out a FIT count. The It's designed to be a fairly simple survey procedure that people can pick up on. You do need to have reasonably warm weather, so I'm not sure that I could really encourage you to all go out and do fit counts at the end of the conference today, because certainly where I am, it feels rather too chilly today. But as soon as we do start to get into slightly warmer conditions, it's fine to do a fit count as soon as we get there. You do need to prepare some way of identifying a 50 by 50 centimetre patch of your target flowers. So you can construct yourself some sort of quadrat using string, or we use gaffer tape to make a sort of flexible quadrat that you can carry around in your pocket. And there's instructions and suggestions as to how you can do that in the documents that are on the Poms website. We encourage you to pick a particular target flower, but if you can't find one of the ones we're suggesting, it's fine to use something different as long as you record what it is. And you focus on that particular flower within your 50 centimetres and count all the insects that actually land on the flowers within the 10 minutes that you use for the count. Having done that, the results can go onto the website, or as of this year, we now have an app available for the fit counts as well. And I'll show you some examples of that in a moment. So as I say, the fit count can be done wherever you can find flowers and suitable weather, and all the information that you need is up on the website, including some quite detailed identification guides. We don't ask people to identify insects to species level for this survey. It is focusing more on the numbers of individual insects, but we do ask you to identify them down to insect group level, and there is guidance both on the website and within the app as to how you can distinguish the insect groups. A lot of them are quite straightforward to distinguish, but there are some quite tricky ones, such as some of the smaller bees and wasps, that do take a bit of practice to get really comfortable with. The Poms project, the Poms surveys, started as a sort of pilot year in the middle of 2017, and then we've had a more complete set of counts for the following years, and as you can see, the numbers of people participating have grown each year. Last year in 2020, we did have a very welcome increase in the fit counts being sent in, and that did seem to be, at least in part, related to the lockdown situation and the COVID restrictions, where people weren't able to get out and about and perhaps go out and visit some of the places that they would normally be recording in. But because the fit count could be done in gardens and very local to your home, and also, of course, because we had such wonderfully warm and sunny weather last April, we had a real boost of counts being submitted, which was very welcome. And we hope we're going to be able to build on that this year. Counts have started coming in, but of course, there have been rather few days in April so far that have actually been suitable weather conditions for this to happen. But thanks to everybody who has already started sending your counts in. And please do send more as it warms up. The results from all this are from the first 4 years are currently being worked on, and there will be some publications from this coming out later this year. But we're also trying to build up a number of additional sort of feedback resources, which will start to appear on the website, including these charts which show the average number of insects per count for the different target flowers. So, for instance, on the left here, we have dandelions, which are one of our target flowers, and obviously something that's around at the moment. And if you spend 10 minutes watching insects on dandelions, on average, you will see 4 insects land within your 10 minutes. You might get a lot more than that. You might get 0 insects, but on average, it works out at 4 per 10 minutes. And the different colours and symbols here show you which insect groups are most frequently recorded. The big grey block for dandelion is the category for tiny insects, and you do tend to get a lot of pollen beetles on dandelions, which are really small beetles that we don't ask you to sort of pick out to group. They just get counted as small insects, and there are a lot of those, but also quite a lot of flies, and solitary bees do reasonably well on dandelions. And on the right here, by contrast, white dead nettle, which is also flowering now and also has an average of 4 insects per 10-minute count, but a lot more bumblebees. Bumblebees really love to get into the white dead nettle flowers and stick their heads deep into the tube of the flower, as shown in the photo at the top there. couple more examples of flowers that are out now or very soon will be. Buttercups on the left there, which don't do so well for bees, but do attract a lot of flies and some of the smaller insects. And hawthorn has a sort of wider range, if you like, with more of the bees represented. Honeybees do particularly well for hawthorn. Poms is not really aiming to monitor honeybees. We are more interested in the wild bees, but obviously people do see honeybees landing on flowers, and they are one of the categories that we ask you to count when you do the fit count. We've gathered a bit of feedback from people taking part in the fit counts, and encouragingly, most people seem to find that it's enjoyable, and most people seem to find that it's not too difficult a survey to do. What's always difficult to get feedback from, of course, is the people who haven't done a fit count and why they have not chosen to get involved. There's certainly more work to do to find out how we can really get people more engaged with taking part in this survey, but some encouraging signs there, certainly. Okay, so as we've seen, the fit counts are designed to be reasonably quick, reasonably easy, can be done in lots of different places. The other survey that Poms runs is a more structured survey and a slightly more complex survey. And this is based around a network of 75 1-kilometre survey squares across the UK now. And it is designed to gather systematic data, and to do that it uses pan traps, which are traps that kill the insects that fall into them. So it is a very different sort of survey approach, and it has been designed so that it catches the minimum number of individuals that we think we need to generate reliable data on country level, UK level changes in pollinator groups. All the insects that get trapped are sent back to the UKCH laboratories and are identified to group and counted, and the bees and hoverflies then go on further to be identified down to species level, and all of that material is stored and is open for potentially use in other research in future. We're working with the Natural History Museum on DNA analysis approaches to try to extract the maximum information that we can from this trapping survey. But it is, again, very dependent on volunteers helping us to do the survey. The 75 squares are available for adoption, if you like, and over half of them have been adopted by volunteers who get an initial training session from UKCH, from the Poms team, who set them up with all the survey kit, provide all the information about access to the square and the trap locations, and having gone through that setup process, the volunteers then carry on the survey, which is carried out 4 times during the summer, once each in May, June, July and August. And the survey actually involves going out to the square and setting out 5 of these pan trap stations in a more or less diagonal line across the square. They're set out in the morning, left for 6 hours, picked up again at the end of the day. The specimens are carefully tipped into tubes and posted back to UKCH. And there's some recording of the flowers, of the plants actually in flower at each of the trap stations. And the volunteers also carry out fit counts within the 1 kilometre square during the middle part of the day when the traps are out there doing their stuff. So it is, as I say, a more involved process. It does involve going out and collecting specimens, which we know not everybody will want to be involved with. But again, we've had some really fantastic volunteers take up this survey for us. This is the current allocation of the squares. The ones in red are the ones where we are still looking for people to adopt those squares. And if anybody's interested, of course, or know any organisations or individuals that might be near to those squares who might be interested, then please do get in touch with us. It would be absolutely fantastic to get more of these taken on if we possibly can. But for the blue squares, where we do have volunteers, quite a few of those volunteers have been in place for 3 or 4 years of the survey now, and we're really very, very grateful to them for enabling this to happen. Poms has been going for 3.5 years, which is still fairly a short time period in terms of long-term monitoring schemes, but the data is now beginning to accumulate and more analysis is being done on it. Even from the initial testing stages, the information that we've been gathering through Poms has started to feed into various publications, and I'm not going to go, I don't have time to go into these in any detail today, but as you might expect, Poms is very much hoping that we are gathering data that is of use to research and to conservation research and development, and the information is going into that route, and we can, if anyone's interested in that side of things, we can obviously provide you with the links and so on. Elsewhere, the Poms methodology, and in particular fit counts, are being picked up by a number of other projects, and we're very grateful for a wide range of organisations for taking this on and working with us. Field Studies Council, for instance, one of the recent Discovering Bees projects is getting people to go out and do fit counts as part of that, which is fantastic. Last year, we got involved in Jimmy's Big Bee Rescue, which some of you may have seen on Channel 4 last year, where they got a whole lot of residents in Peterborough to get involved with conserving volunteers in their open spaces, doing some of the things similar to the ones that Jodie was outlining in the previous talk, in terms of trying to improve the habitats for pollinators and using the fit count methodology to start monitoring the results of that. And just a couple of weeks ago, the cross-pollination project launched, which is working in the UK and internationally, has a particular focus on working with schools, but other people can get involved as well. And they're doing a really fantastic range of activities, including the fit count based survey methodology to feed into that. Beyond the UK, the fit counts are also proving popular around the world, in fact, and there are a number of projects now that are picking up on the methodology, and obviously it has to be adapted for the different types of pollinator. Unfortunately, we don't get to count hummingbirds in this country, but in Chile they do, and in some of the other countries there's some really interesting work going on using these same methods. So we're really pleased to see that the methodology does seem to be attracting interest and could potentially build up to something that's giving more information about changes in pollinators, particularly in pollinator abundance in many parts of the world. Okay, so we have a website, a new Poms website that is coming very soon. On the 20th of May is when it should happen. And if it doesn't happen, I'm certainly going to be in trouble, but that's what we're aiming for. And that will have all the information. For now, you can find all the information on the UKCH website, but it will all transfer over in just over a week's time. What you can do as of about a week ago is to download the new Fit Count app. It's in the app stores for both Apple phones and Android phones. And you can take this out in the field with you, and it has all the information to actually do your count on the app in the field. You can then upload it either from the field or when you get back home. And that information all comes into Poms and forms part of this. So please do spread the word that that is available for everybody to use. So hopefully these messages have come across. Please do consider taking part, and particularly with the fit counts. It's one of those surveys that really does benefit from as many people taking part as possible. So please spread the word about that. The 1 kilometre square surveys are there. If you're interested and happen to live near one of the squares, that would be fantastic. But this is not a replacement for traditional hoverfly and bee and butterfly and everything else recording. The recording schemes are still doing that fantastic job, and that is part of the data that's being analysed. So this is an add-on, not a replacement. And as I say, the 1 kilometre squares are not really, none of them are in urban areas. It is designed not to cover urban areas, but the fit counts do cover urban areas. And the picture there shows an event from Bees Needs Week a couple of years ago with the Mission Invertebrate project in London's Green Park, and it is something that very readily can be done in urban areas, and I think does hold a lot of potential for getting people involved and for getting people just to take more interest and learn a bit more about pollinators. So that's the end of the presentation. Thanks as always to the project partners and funders, and particularly to our wonderful volunteers and the landowners who give us permission to run these surveys. So please get in touch if you'd like to know more. And if we have any time for questions, I'm very happy to answer a few questions.
Thank you very much indeed, Martin. Absolutely huge response in the chat to your talk. I'm just going to pick up out a few questions in the small time we have. One question that came through from Paul Seligman, can you use an equivalent area to the 50 by 50? So could you have as long as the area is constant, could you have a long thin area or even a circular area with the same area? So,
that's a good question. The protocol for the fit count says that it has to be 50 by 50. We don't go out and check it down to the last millimetre, of course, and with the sort of slightly flexible quadrats that we tend to use, then yeah, it's not an exact square necessarily, but we don't really suggest that you use a long thin strip, but within that 50 by 50, you're only focusing on one particular type of plant. So you don't have to include all the plants in the area. You can, so if you have got a linear strip, you can still imagine your 50 by 50 square on top of that linear strip and just focus on the plants within your square. You don't need, as long as you're counting the numbers of flowers, which we ask you to do, that's absolutely fine.
Okay, Sarah Patton and Hannah Fullerton reported some problems with the app. So if you could get back to them in the chat, that'd be really good.
Okay. Best thing to do with that is to send any feedback into the Poms email address, and we will pick it up from there. But I will have a look at the chat as well, certainly. And
Victoria Burton asked, do you have any information about the background of the people who have taken up the 1 kilometre squares?
Not in any systematic sense. We don't collect any sort of demographic information on the people taking part. We're just very grateful for anybody who's happy to support it. So I'm not quite sure what Victoria's getting at there. We obviously do know informally because we do go out and meet these people, so we do get to find out a little bit about them. And they have come from a variety of backgrounds. Some of them have just contacted us and said they're interested. Some of them have come about through contacts with the local record centre in that area. So there's lots of different ways that people have got involved. So yeah, that's probably all I can say on that, really.
And Stephen Green asked, how are you defining buttercups?
As the plants that are commonly called buttercups. So again, we don't ask people to identify them to species. So that several of the so-called target flowers are really aggregates. So thistles are a target flower, but we don't ask people to identify exactly which thistle they're using. They can do if they know it, they can record it, but it's not essential to take part. And we do ask people to submit a photo of the flower that they are using, and those photos are checked as part of our analysis process. So we are able, in most cases, to confirm what they are, but in terms of, you don't actually need to know exactly which buttercup you're looking at, but hopefully it is a buttercup, and if it isn't, we do check on that.
Okay, and there's also a really interesting question from Nick Ferraby, but I think it's too long for us to take here, so maybe you could pick it up in the chat, Martin. Thank you very much indeed.